Welcome everyone to the recorded webinar series on LED-based photoacoustic imaging. My name is Mithun and I'm a Research and Business Development Manager at Cyberdyne. In this series, several key opinion leaders in the field of photoacoustics will talk about LED-based photoacoustic imaging and its preclinical and clinical applications. Today's very exciting talk will be about LED-based photoacoustic imaging of human inflammatory arthritis and will be delivered by Professor Xueding Wang from University of Michigan. Before working as an independent principal investigator, Dr. Wang received his Ph.D. at Texas A&M University and postdoctoral training at the University of Michigan. Dr. Wang has extensive experience in development of medical imaging and treatment technologies, especially those involving light and ultrasound. Sponsored by NIH, NSF, DoD, and other funding agencies, Dr. Wang's research has led to over 130 plus peer-reviewed journal papers. Dr. Wang was the recipient of the Sontag Foundation Fellow of the Arthritis National Research Foundation in 2005, the Distinguished Investigator Award of the Academy of Radiology Research in 2013, and also was elected to the Fellow of AIMBE in 2020. He is also sitting on the editorial boards of scientific journals including Photoacoustics, Ultrasonic Imaging, Journal of Biomedical Optics, Medical Physics, and Nature Scientific Reports. In this webinar, he will talk about the potential of LED-based photoacoustic imaging in detection and staging of inflammatory arthritis in human subjects. With that, I will turn it over to Professor Wang.
Okay, so my name is Xueding Wang. I'm a professor with the University of Michigan, Department of Biomedical Engineering. Today I'm going to introduce you our work on the LED-based photoacoustic imaging for detection and staging of inflammatory arthritis. And this work is in collaboration with the Department of Radiology and the Division of Rheumatology at the University of Michigan. I am a big fan of optical imaging because really optical imaging has many advantages. And on this slide, I show you several example images from optical imaging modalities. On the left side, you can see an optical coherence tomography image of a human retina. And at below, there's confocal microscopy images of some neurons. And this type of optical images can give you a lot of detailed structure informations of biological samples. However, this type of images, although have a high resolution, the excellent spatial resolution can only be achieved in very superficial tissue with imaging depths about like a millimeter or hundreds of microns. Well, if we want to use light to see tissues deeper, for example, if we want to use the diffuse optical tomography to image a human breast cancer, and you can see here the image quality is downgraded very quickly. Okay, and on the lower side, this is optical computed tomography. Okay, for a human PIP joint. Okay, it's a cross section of a human finger joint. Again, if we want to use light to see deeper, the image quality will degrade very quickly. You can still see some structures, however, the spatial resolution is very limited. And the reason of that is the very strong optical scattering in biological tissues. There's new technology invented. It's called photoacoustic imaging. And one of example technology is called photoacoustic tomography. For example, if we want to image the brain of these small animals, we will use nanosecond laser pulses to illuminate this animal head, and the light will penetrate through the skin and skull into brain tissues and will generate the ultrasound waves due to the thermoelastic expansion. And this type of ultrasound signals can be detected out of the sample by using ultrasonic transducers, we can also position the ultrasonic transducer around the sample or use a transducer array to receive the signals from all the directions. And then by performing image reconstruction, we will be able to get the image of this sample. Here, this is an image of a mouse brain, which is obtained through the skin and the skull non-invasively. So it's a totally non-invasive imaging technology. And this photoacoustic imaging, by using laser-induced ultrasound, this photoacoustic imaging can combine the very good optical contrast with excellent ultrasonic resolution. So the photoacoustic imaging is not suffered from the overwhelming optical scattering in tissues anymore. It can show you the highly sensitive optical information in deep tissues with excellent spatial resolution. And at Michigan, we have been pushing the photoacoustic imaging application to different diseases. Okay, so our lab is in the medical school, and one of our clinical applications is arthritis. Okay, so you may ask me, why are we interested in arthritis? Arthritis is a huge problem. Okay, and in United States, every year the cost on arthritis is over 65 billion. Okay, and over 46 million Americans are currently suffering from arthritis. One of the arthritis is called rheumatoid arthritis. It's hitting the human finger joints, peripheral joints. And another one is called psoriatic arthritis. It's not only hitting the joints, but also the tendons. So how photoacoustic can potentially contribute to rheumatology clinic? So let's look at the progression of inflammatory arthritis. Usually, the arthritis starts from the inflammatory molecular markers in tissues. You can see a lot of molecular changes in soft tissues. And then later, you will notice some functional changes in the human tendons, in human joints. For example, you can see that the joint is fat and red. Okay, it's a big fat joint, and that's due to the hyperemia, which is the enhanced blood volume. And also, you very often can see the blood oxygen saturation is decreased. It's called hypoxia. And that's all reflecting the metabolic activity in the arthritic joints is different from that in the normal joints. And later, we can notice some anatomical changes. For example, synovium thickening, the fusion, the bone erosion. Okay, that's kind of happened at the late stage of arthritis. The current imaging modalities, for example, CT, MRI, ultrasound, they are all focused on the anatomical changes in the joints. Okay, so we understand that when this anatomical changes happens, it's relatively late. Okay, this type of changes are mostly irreversible, so the treatment effects could be much worse. So what we are hoping to do is we can detect the arthritis at earlier stage of the arthritis progress. So if we can detect these functional changes in the joints by using photoacoustic imaging, we will be able to start the treatment earlier. We will also be able to monitor the patient's response to treatments much quicker. Okay. Of course, another potential application of photoacoustic to arthritis is to do this contrast enhanced photoacoustic imaging so that we can even diagnose the arthritis at the molecular level. So we can see the molecular markers in the tissues. But that's not the focus of today's talk. So again, today's talk will be focused on the label-free photoacoustic imaging of arthritis for detection of functional changes in human finger joints. So at Michigan Medical School, we have started a clinical study on photoacoustic imaging of arthritis, and this is the very first clinical study on arthritis. And our goal is to involve over 100 patients with arthritis, and our goal is to validate that photoacoustic offers a new tool for rheumatology. On this slide, we this is our lab built ultrasound and photoacoustic dual modality imaging system for arthritis, and this system was built on a Verisonics ultrasound imaging platform. Okay, it's a commercial research imaging platform, and this system is working with a class 4 laser. It's a very big laser system. It's a tunable laser. When we try to image a human finger joint, like on the right side, we can put an ultrasound probe and we can put optical fibers and integrate them together so that we will be able to get ultrasound images from a finger joint. At the same time, we can get a photoacoustic images. Here is an example of ultrasound B-scan grayscale image from a human finger joint. And on the right side is the photoacoustic image from the same patients, from the same human subjects. As you can see that the photoacoustic and ultrasound really can present you similar tissue structures. But the goal here is not to present you the structural information in the tissue. The goal here is to, by leveraging the highly sensitive optical contrast, we will be able to show you some functional changes as a result of inflammation. Here is an example result of ultrasound and photoacoustic dual modality imaging of inflammatory human MCP joint in vivo. This one is the grayscale ultrasound image, and on the right side is the grayscale photoacoustic image. As you can see that, in the photoacoustic and ultrasound, there are a lot of similar structures. However, in the photoacoustic image, we can see a lot of strong signals surrounding the joint bone. And this type of strong signals, what we call hyperemia, or say, in other words, enhanced blood flow due to the inflammation. Okay, we can now combine the ultrasound image and the photoacoustic image together as shown in this combined image, and we use ultrasound, grayscale ultrasound to show you the structural information, and the pseudo-color photoacoustic image show you the hyperemia information. Okay, this type of combined image is very much similar to the gold standard Doppler ultrasound. The difference here is in the Doppler ultrasound, what we are looking at is the blood flow, okay, and in photoacoustic image, what we're looking at is the enhanced blood volume. So the mechanism is different. We can both detect the active joint blood flow, but the photoacoustic image is not relying on the speed of the flowing blood. That's why the photoacoustic image can detect the enhanced blood flow as a result of inflammation much more sensitive. Here is example results from a normal volunteer, and in this combined image, you are not be able to detect the enhanced blood flow because, again, this is a healthy joint. We scanned many patients with arthritis, as shown here as the example results. A lot of normal volunteers. Photoacoustic image, we cannot just detect inflammation, but more importantly, we can quantitatively evaluate the joint arthritis activity. So in order to evaluate the hyperemia quantitatively, we can quantify two parameters in the photoacoustic image. The first one, we quantify the density of the colored pixels reflecting the hyperemia. Okay, and the second one is the average intensity of these colored pixels. Both of them can reflect the inflammation. As you can see in the statistic study, we compared the measurements from the arthritic joints to the measurements from the normal volunteers. As you can see here, the colored pixel density in the arthritic joints is much higher, and the average intensity of color pixels also much higher in the arthritic joints compared to the normal. Okay, both of them can differentiate the arthritic joints from the normal joints with a statistic significant difference. And so another parameter we can measure, we can quantify by using photoacoustic imaging is the blood oxygen saturation. Okay, that's depending on the different optical absorption spectrums of two types of hemoglobin in the tissue. One is the oxyhemoglobin, the other one is the deoxyhemoglobin. So because these two types of hemoglobins have different optical absorption, we can use more than 2 different optical wavelengths to image the same joint, and so that we can quantify the blood oxygen saturation in the joint. So we have studied both the arthritic joints and the normal joints. As shown here, the left one shows a blood SO2 image in an arthritic joint, and the right one shows the blood SO2 image in a normal joint. Okay, so you can see clearly the SO2 level in the arthritic joint is slightly lower than the normal joint. We have also performed statistic studies for the arthritis group and the normal group. As you can see, then the photoacoustic image is able to differentiate the two groups by measuring the blood oxygen saturation in the joints. The second second parameter for us to differentiate the arthritic joints from the normal joints. So we have already pushed this technology and this application in rheumatology clinic for several years. And as I mentioned, we collaborate closely with the rheumatology division, rheumatology clinic at the University of Michigan. So the rheumatologists, they told us after collaborating with us for many years, they told us some challenges when using the photoacoustic imaging to scan the human joints to diagnose the inflammatory arthritis. So one of the challenge here is the high cost, of course, right? Because of the high cost involving the class 4 lasers, we have only one system sitting at the University of Michigan, and we cannot perform really multi-sites clinical study. And another problem, which is a little bit bigger problem, is the large footprint of our photoacoustic imaging systems. Again, we're using a large class 4 lasers, so our system cannot fit into the small rheumatology clinical rooms. We cannot perform the clinical study very conveniently. We have to bring the patients to our lab to scan them. And also the system is difficult for use. We have to train the rheumatologist, the radiologist, how to take care of the lasers, how to warm up the lasers before they can start to scan the patients. Of course, the safety concerns of class 4 laser is also a problem. The rheumatologist, radiologist, and our technicians, when they use the system, they have to put the goggles on before they can, turn on the laser. And that also makes the patients and the operator not very friendly, because the patients also needs to wear the goggles during the scan of their fingers. Later, we have been thinking about any potential alternative light sources that we can use for photoacoustic imaging of arthritis. A candidate light source is the LED light source, we have noticed, and another one is the diodes laser. Both one lower in cost and also very convenient for use. And later, we have collaborated with Cyberdyne Corporation. And so Cyberdyne Corporation is a company who building the Acousticx, which is LED-based photoacoustic imaging systems. And the system is also providing not just the photoacoustic imaging function, but also ultrasound imaging function at the same time. So when we try to image a human finger joint, as you can see here, right? So the ultrasound probe can be put onto the finger, and at the two sides of the probe, we have two LED bars, which can provide the light that used for photoacoustic imaging of the human joints. For this system, you can notice a lot of advantages for potential applications to rheumatology clinics. Clearly, the system is much lower in cost because we don't have to spend a lot of money on the class 4 lasers. The LED light sources are much cheaper. And also, more importantly, this system has a much smaller footprint. So it's much smaller device, and we can put them in the rheumatology clinic rooms very, very easily. Okay, and it's very easy to use. It's just turn on and then ready for use system. And our rheumatology and the radiology collaborators, they are both very much like the system because they can use it very easily, and also give you much better safety because there's no very strong class 4 lasers anymore, and the LED light sources are much safer. And it's super patient and operator friendly. So our patients involved in the study, they don't have any concerns by putting a LED light source on their fingers to do the imaging. So really, the LED-based photoacoustic imaging offers a point-of-care tool for clinical screening and the treatment assessments of inflammatory arthritis. And here, before we start to use the system on our arthritis patients, we first try to evaluate the system performance by scanning some normal volunteers. And in this study, we scanned a finger joint by using this system. So what we did is a three-dimensional scan by scanning along this finger. And so by doing a 3D scan of the finger, we can show the vasculature in this finger along each view. This is the coronal view, this is the axial view, and this is the sagittal view. And we can also show the vasculatures in the fingers along a sagittal projection view. You can see a lot of vessels in this finger. They are all imaged successfully. So, another function that can be provided by this LED-based system is the dual wavelength imaging of blood oxygen saturation. So the LED source, they are not tunable. They are not like lasers. However, they can also provide you more than one wavelength. So for example, over here, this one shows you a dual wavelength LED bars. So, and this dual LED bars works at 690 and 850 at the same time. So by working at this two wavelengths, we are able to measure the blood oxygen saturation in the fingers. So this is a study also on a normal volunteer. Okay, you can see this is a human finger, and we are able to detect some vessels. Okay, and then this normal volunteer, he is he's trying to change the blood oxygen saturation by holding the breath. Okay, so by doing so, he was able to put the blood oxygen saturation at different levels. Okay, and the y-axis shows the photoacoustic measurements by using the dual wavelength LED-based photoacoustic imaging systems, and the x-axis shows the blood oxygen saturation measurements from a pulse oximeter used as a gold standard. And as you can see, the photoacoustic imaging by using the LED-based system has a very good match with the readouts from the pulse oximeter. So now we have been using this LED-based imaging system on arthritis patients. So here is example results from this imaging system. The grayscale is the ultrasound image showing the tissue structures, and the pseudo color, this kind of pinky pixels, are from the photoacoustic images. As you can see, this is an image from an arthritic joint. As you can see, the hyperemia is able to be presented very, very clearly. And this image has a good match with the Doppler ultrasound images from another system. This is a commercial ultrasound system. And our photoacoustic image has a good match with the Doppler ultrasound for detecting the hyperemia. So here are some example images from the patients with clinically active arthritis. In each one, we are able to detect the hyperemia in their finger joints. Example images from normal volunteers. And because they don't have active inflammation, we're not able to detect a lot of activities in their finger joints. So the photoacoustic image, as I mentioned, is able to detect the arthritis, but more importantly, it can help us to stage the severity of arthritis. So here, this result is from a subclinically active arthritis. So the meaning for subclinical active arthritis is that this patient is recruited by the rheumatology clinic, and to confirm that they do have inflammation in their finger joints. However, as you can see in the left image, we cannot detect the inflammation by using ultrasound. That's because this patient has already started some treatment, so their joint activities are not very high. So there's no active flow in ultrasound. Okay, but when we scan these patients with the LED-based photoacoustic imaging system, we still can pick some inflammation in their finger joints. So this one is example results to show that the photoacoustic technology is more sensitive than Doppler ultrasound in picking up this very mild inflammation in human finger joints. Okay, so we have really three groups of patients. We have patients that are clinically active with clinically active arthritis. Okay, and for these patients, we can detect their inflammation in the joints by either photoacoustic or by ultrasound. Both of them are able to detect their inflammation. We also have a group of patients which are subclinically active arthritis, and the ultrasound are not very sensitive to detect the inflammation. However, photoacoustic can still detect the active inflammation in the joints. We also have a group of normal volunteers. Okay, and with the photoacoustic imaging results from the three, quantitatively evaluated their activities in their finger joints. So we also quantified the two parameters. The first parameter is the density of the colored pixels in the joints. The other parameter is the average intensity of the colored pixels in the joints. And for each group, we have the measurements here, right? As you can see here, this is the clinically active arthritic group, and this is the subclinically active arthritis group, and this is the normal control. And by performing t-test, we're able to differentiate any of the two groups easily. Okay, and by using another parameter, we can also differentiate the three groups. So this one tells you that the photoacoustic imaging is not just detect arthritis, but more importantly, can quantitatively evaluate arthritis. Let's have a very quick summary. So the photoacoustic imaging, by working with a single wavelength, is capable of detecting the hyperemia as an important biomarker of human inflammatory arthritis. And the photoacoustic imaging can also work with more than two wavelengths and can detect the hypoxia as another biomarker of human inflammatory arthritis. And photoacoustic imaging based on LED as the light source can provide sufficient depth and sensitivity in mapping the microvasculature and inflammation in human finger joints. And the LED-based photoacoustic imaging offers a practical and a point-of-care device for clinical management of inflammatory arthritis. All what I want to present. I want to thank you very much.